Thank you very much for the invitation, and thanks very much for joining us this evening. It's a nice, cold, wintry day, but it's always nice to be talking about canola, and especially during a growing season that's yet to be. So today I have the pleasure of talking about some of our results from our annual canola survey. I also want to branch out into some of the other interesting things we've discovered as part of some of that work that we do in canola. I also want to just start off by acknowledging co-authors here, which should be my technician, Shelby Dufton, Sasha Chisholm, who's provided some data for weather, Dr. Kelly Turkington, who's also provided some data for weather, and then Dr. Megan Van Kosky. And Megan and I have been working with the Prairie Pest Monitoring Network, and this is, of course, a large project that basically funds a lot of our canola surveying work. So very important co-authors. And of course, this is our very nice backdrop. Maybe not the luscious field of canola, but this is the type of thing that we do each year. It's one of the big projects we do in our program at Beaverlodge. And of course, our canola survey definitely is one of our big feature projects. So today I'm going to be talking a little bit about everything. Oh, we'll see if interesting. There we go. We're advancing slides now. We're on track. So just a bit of background in terms of the pest management program. I've been at Beaverlodge since April of 1999. And so it's not just the hair color, it's actually been quite a few years. Our program usually ranges anywhere from field plot research. Our annual canola survey is a big portion. We've done a lot of different projects, and so some of the stuff I'm talking about today would be covered by some of our environmental stewardship monitoring project that we just finished. And we're also going to touch really quickly on the Peace Region Living Lab Ecosystem Services activity, because some of that work has been precipitated by what we've been doing in our annual canola survey. So these are some of the critters that our program is very well versed in. I think over the many years that I've been at Beaverlodge, we've had the benefit of working in several annual crops like canola, wheat, a little bit in peas, but we've also had major funding through the Peace Region Forage Seed Association that allowed us to do a lot of work in soil and the canopy of our perennial grass and legume seed production systems. So here's a small smattering of probably some of our biggest, probably the most prominent, more urgent insects that we're dealing with at present. Of course, we actually have about 45 species that our program has actually been monitoring, and that's quite a long list, but it's quite a big region. So the annual canola survey actually fits into a much larger project, which for many years has been funded under the umbrella of the Prairie Pest Monitoring Network. At present, we're now funded under a cluster activity. You can see our stakeholders are listed here, but essentially you can see we're part of a much wider network that spans across the Canadian prairies. This is actually the 2024 survey stops for all of this project. And so as you can see, the different colored dots actually correspond to different insect pests that are monitored throughout the growing season. And you can kind of see here in the Peace River region, our little chunk of the woods, if you will. That's primarily a lot of work done by our program, but there's also a lot of contributors through Alberta Agriculture, and of course our regional networks that are affiliated with Alberta Agriculture and Irrigation. And then also there are some key stakeholders who actually do some monitoring. So we can't take credit for all of those dots, but there's certainly a number of dots that are associated with our annual canola survey. It's quite a large network, and as you can see by some of these numbers, it's really a very vast undertaking. And in fact, I think globally, it's still one of the largest networks that covers the greatest number of arable acres in the world. So we're going to be talking about the annual canola survey. I'm giving you a little bit of a preview here of our 2025 data. But again, just for stakeholders, the various insects that we're capturing are they basically arise through sweep net collecting. So this is assessing what's in the canola canopy. And we do it over a very short period of time, relatively speaking. And we also do it in canola that is in commercial sized fields. Again, this is a little bit unusual compared to some of my other colleagues who are doing smaller areas and perhaps not able to focus primarily on commercial field crops. A little picture of our team from this past summer. It does take a huge number of people to get this done. We do target the early to mid-flowering period of canola production. And so it's a very vast undertaking that involves a lot of people across the entire region, a number of people that we even call in other areas to see what the weather's like so that we can get in and out of fields. And of course, we do have a very large permission to enter form that now needs to be filled out prior to the survey. I also want to really mention the fact that we have a lot of networking with Agriculture and Agri-Food Canada in terms of pulling together some data for weather and temperature and precipitation specifically. And then, of course, we are kind of piggybacking on some of our provincial networks, and those would be folks like the BC Ministry or the BC Pest Monitoring Contractor in the BC piece, and of course, Alberta Agriculture and Irrigation. So a large group of people, and I'm going to take you back a little bit because this is really one of the big reasons that we started the annual canola survey. Way back when, there was this thing called cabbage seed pod weevil, and you can see in 2002, it was still a relatively new invasive species to Western Canada. The areas in red and orange and yellow actually indicate where the cabbage seed pod weevil first started and where it was first detected in Western Canada. And it was at about this point that we started to have some conversations about detection in the Peace River region. We were very concerned about how quickly this canola pest might spread throughout Western Canada and what was its potential for moving north. And of course, we were already doing a lot of work with lygus in canola. So we had a bit of experience with sweep net collecting in canola, had a bit of an understanding of how canola grows in the piece too. But this map really served as a large impetus for what we have now been doing for 23 years. When we flash forward to the cabbage seed pod weevil distribution maps now for 2025, we can see how that invasive species has spread across Western Canada. We can see some very high populations. Again, the primary point of introduction was in southern Alberta, just south of Lethbridge, Alberta, and we can see how far north it has spread so far. I'm really pleased to tell you that the one thing that we have discovered is all of our annual surveying, which would be represented by these individual circles. We have never managed to find cabbage seed pod weevil in any of those samples. So that's a really encouraging situation. And actually, there was a publication that just came out this past year that actually documented the distribution of cabbage seed pod weevil and its predicted spread in the future. So that data set is really important because not only are we corroborating that there are no cabbage seed pod weevil, we've actually been able to establish a 23-year period where we've never been able to find it in our canola. Of course, now that I say this, I'm very worried that I'm going to jinx us for the future. But this is the current state of affairs across Western Canada in relation to cabbage seed pod weevil. So one of the things I wanted to do today is kind of hit some of the highlights and some of the things that have been, first of all, how the canola survey has developed over the many years that we've done it, and then also look at some of the segues that we've made, because there have been some pretty interesting things that we've discovered along the way that we never necessarily started out looking for. One of these things for the piece, what we're looking at here is the lygus densities in canola. Now, this is our 2025 map. And in fact, it's not really too exciting in a lot of ways because we're not seeing big hotspots. In fact, we're seeing very low populations of lygus. So not only are we looking at the adults and the nymphs that you'll see in the top left-hand portion of your screen, but we actually have started to develop this survey over the last almost 15 years. It's starting to be a very large biodiversity study. We initially targeted our biggest economic insect pests, but over the last 15 years, we've been able to develop that data set through expertise in our own lab, with our technicians and myself. We've been able to kind of look at what is the biodiversity in the canola canopy. It's turning out to be a very large undertaking with almost 140 taxon units. And that actually is starting to be a really interesting representation of how diverse the insects can be in your canola fields. And in fact, the piece is actually very fortunate because not only have we enjoyed some fairly low insect pest pressure from Lygus, we've actually seen some very unusual bees. And we also think that there's a lot more going on for pollination in terms of flies. And some of these samples are going to help us demonstrate, first of all, what the diversity is, how things change, and how things change over time, and with cropping practices. So one of the big things that we've done with the canola survey, we usually target going in and out of commercial fields in about 11 minutes. That sounds pretty quick, but we don't usually need to be doing all that much. We try to condense it into a very short period so that we're able to sample, do the biosanitation for our vehicles and our equipment, but we're also starting to look at the surface stubble. The piece has a very bad reputation of this canola, snow, canola rotation. And so very early on, almost 12 years ago, we decided that we would be able to actually take a quick look at the surface stubble and actually start to document what was underneath the canola. How much canola on canola was there actually out there? The results have been a little surprising. The first couple years we started this, almost 12 years ago, there was some canola on canola, but you can see in 2025, there's relatively few canola on canola fields, at least compared to the 121 fields that we sampled. In fact, what we do see, and we've been able to kind of document, is this very tight canola wheat canola rotation. So you can see in 2025, the predominant surface stubble under our flowering canola was actually wheat. Now this becomes a really important story as we move forward. So just keep that in mind. Canola, wheat, canola seems to be a very common rotation for the fields that we're surveying in. So I mentioned the level of biodiversity. This is really just a small diagram. When I first came to Beaverlodge, I had the privilege of working with Lygus and canola in southern Alberta. And so from that work, I had a fairly good understanding of how many generations, how that might change as we move further north, and how tightly associated Lygus was with canola. Since I've been in the piece, the canola survey has served to actually document much more variation in the number of insect species that are utilizing the canopy. We have very complex interactions, so it's not just an insect pest and a host crop. In fact, we have 3 different levels of trophic interactions. So in fact, we have Lygus, which utilizes the canola in the background, but we have, of course, some of these general predators that are actually predominantly picking out and feeding on some of the more immature Lygus nymphs. Some of those general predators will feed on Lygus adults, but we've actually done some lab studies that indicate that, in fact, they really don't enjoy adults as much as they do nymphs. We also have discovered that there are some very important parasitoid interactions. So very early on, we were finding with cabbage root maggot, we were easily finding this nice little grove beetle, which is Aleochera bilineata. It actually is very common in your canola fields because it's actually a parasite and a predator of cabbage root maggots. And in fact, the adults will actually overwinter inside the cabbage root maggot case, which is what you're seeing here. That's in fact a newly emerged adult. So a very tight association, even though it's not Lygus, it is a tight association with another insect pest that we commonly find. And of course, those little cabbage root maggot, Puparia, overwinter in the fields. One of the other things that have been really important is that we very early on, after the first real outbreak of wheat midge in 2011, it wasn't long after that, I think it was about 3 years afterwards, we started to find huge numbers of these tiny little parasitic wasps. These individuals are maybe at best 2 millimeters long, but we were finding them in the canola. And I'm going to come back to this because it was a really important finding. But at the time, we were finding huge numbers and really starting to scratch our head because we were not sure what they were and where they were coming from. But essentially, what we've got here is a diagram of how Lygus interacts with its host crop, some of the other trophic interactions with some of its general predators and some of its parasitoids, and it's really demonstrating to us how complex the canola canopy can be. And this isn't even including any of the pollinators that we know can be in that crop. Now, one thing I am doing today is I do have a QR code at the very end, and I know this presentation is being recorded, but for those of you that have actually taken the time to join us today, I have included some sheets or slides that have hyperlinks in, and they're intended to help you out with some scouting tips, some information on different insect pests that I'm going to be covering. So I'm not going to go through this slide, but it does actually hold some good information and some good hyperlinks, which are these blue areas on the page that you folks can click on with a PDF later on. So I mentioned these nice little tiny wasps. Well, we started to pull apart these sweep net samples, and sure enough, we were able to access some expertise from a taxonomist in Ottawa, and they confirmed what we actually started to suspect. And that is those tiny little 2 millimeter long wasps were actually a beneficial insect. Here he is right here, or here she is, I should say. And in fact, they are Macrogleanes penetrans. It's a proven economically significant, actually, parasitoid that attacks wheat midge. And in fact, that's this organism we're seeing here in the bottom right. So wheat midge, very tiny, nice bright orange. It turned out we were finding Macrogleanes penetrans in some huge high numbers in the canola that was at flower. So while it didn't at first make sense to us, when we actually went back, we were very fortunate to start to collect that surface stubble information, because what we were finding is most of our canola was on wheat stubble. And in fact, these these Macrogleanes were emerging from what was last year's wheat. Okay. They were in such high numbers, and in fact, in 2025, we can actually see a pocket here of Macrogleanes penetrans just over by folair, some very high densities, almost 36 per 10 sweeps. So what we have now started to recognize and communicate to the producers in the region is the fact that they need to be very careful in flowering canola, particularly if it's being grown on wheat stubble, because flowering canola, not only is it a risk for sclerotinia, but we also have these very valuable biocontrol agents that are emerging at the same time. They're actually emerging and trying to find more wheat, and especially more wheat midge. Now, let me just go through the life cycle of wheat midge so that you can hopefully sort of link all these pieces together, because it's a bit of an abstract situation. This is the 1-year life cycle of wheat midge. So in fact, if we start here, right now outside under the snow, we have these cocoons that are in the soil, and they would have formed in what was wheat midge last year, so 2025's wheat. Okay. What happens early in the spring is those cocoons will continue to develop, and if they're parasitized, in fact, what's going to be inside a cocoon is actually a parasitoid. And then, in fact, both the wheat midge and the parasitoid then emerge in the crop, and then they go and seek out and find wheat that's at early anthesis. The big thing here is we know that as the wheat is finished, the wheat midge and the parasitized wheat midge are dropping into the soil, and they actually then overwinter. So this very tight rotation of canola, wheat, and canola is actually very important, because we have Macrogleanes penetrans, that wonderful biocontrol agent, is going to be overwintering in last year's wheat, but it's going to be this year's canola for many of our producers across the region. And that means we need to manage it as it goes into flower very carefully. We don't want to be using insecticide spray if we don't have to, because at flower, some growers we do know will put in their fungicide, and they may throw in some insecticide as a tank mix. Well, this is exactly the situation we want to avoid, because we know that there are Macrogleanes who will be emerging at that same time. And while you might think you're managing some insect pests, what you actually would be doing by doing that tank mix and putting the insecticide if you don't need it, you might actually be jeopardizing your nice beneficial organisms. Well, I'm going to talk very quickly here about wheat midge because there are maps, and I know Amanda Jorgensen is going to be talking about these next week for you folks. But fortunately, for most of the piece, we do know that we have relatively lower populations of wheat midge. However, that doesn't change the fact that we want to preserve and really protect the Macrogleanes penetrans that are out there, because they're like a reservoir or a backup army for when wheat midge populations do start to increase. In fact, those very nice parasitoids respond very quickly to outbreaks like what we're finding in Saskatchewan and have documented with these very high populations indicated in red and orange. So again, I mentioned that I was going to have some quick sheets here, and here's a really good example of the economic threshold for wheat midge, some information on how to monitor them. But we really want to communicate to people, and one of our surprise findings was the fact that a biocontrol agent that was actually functioning in wheat is actually tightly associated with what we're finding in canola at full flower. So it's very interesting and demonstrates how interrelated our field crops can be. Another good example of some of the parasitoids that we've found in some of the canopy sweeps that we do in canola has actually been some of the general parasitoids that attack cutworms. So for many years, we've done a lot of work with cutworms. This was pretty much in the early 2000s. We had a very large CARP funded project to look at cutworms. At that same time, our program was very fortunate to be part of some funding we had from the Peace Region Forest Seed Association. And in fact, what we found at that time with some of the cutworm infestations was very high level of parasitism, somewhere around 30 to 35%, which is relatively high for parasitism in some of these insect pest species. But again, a really good example of how these really tiny wasps actually are utilizing canola. And it's something that we don't always think about. Quite often, we're very concerned about managing insect pests, and of course, for good reason. The other thing that we've started to see, and what we are wanting to do more with, is actually looking at some of our grasshopper parasitoids. So this doesn't really have as much of a tight connection to the canola survey, but one of the things we've discovered with some of our pilot projects with grasshoppers is that there's actually relatively high parasitism of grasshopper eggs. And now we're starting to ask the question, okay, are those showing up in some of our canola survey sweeps? Because we know some of these parasitoids as adults would actually feed on nectar or pollen. So this is now a new project that we're hoping to get more support for, because we're starting to actually also look at the development and the species differentiation between the different grasshopper species that are present across the Peace River region. And it's starting to be quite an interesting story. Well, I think grasshoppers are always a dominant question. And one of the things that we find with canola survey is that we certainly see them in the ditches. We are sometimes seeing them in the canola crop. But really, all in all, we always want producers to be very aware. And while 2025 was actually a higher population year throughout the Peace River region for grasshoppers, some of the work we've been doing with some of that egg stuff and now also the adult stuff is looking at the species differentiation for some of these grasshoppers. And it's kind of spilling over to some of the fieldwork that we do for the canola survey, which allows us to be across the entire Peace River region. We're now actually piggybacking some of our survey samples for grasshoppers at the same time we're out in canola. What that's telling us is it's giving us a bit of an early snapshot of, first of all, what kind of grasshopper nymph populations are out there, the earliest adult indication, and then also some of the species diversity work. So the canola survey has been very important for us because it gets us out and about at some very critical times during the growing season. Some of the work we're doing here is really trying to understand this Bruner's grasshopper. And here's a nice close-up shot that Shelby Dufton's taken for us. This particular species seems to be our pest or high density species in the odd years. What we're finding is that's not necessarily crossing over into the BC piece in the same way. So there are lots of questions about this one particular species, which seems to maybe follow a 2-year life cycle versus our more annual crop species of grasshoppers. Now here's a good example. This is from 2023. What we actually did is we used the canola survey samples taken in July to actually target areas of high populations of grasshoppers, and we went back in August. So this is one of the ways that we're trying to make our fieldwork very efficient. One outing is piggybacking 2 or 3 different sampling efforts, but also serving to help us get early warning signs for what's happening later in the year for other species. Again, here's a nice quick sheet for you folks to also access, and I do want to just highlight the fact that we have in this slide deck also included the thresholds for grasshoppers. Always very handy, always good to have at your fingertips, especially on big grasshopper years, and we're going to hope that next year isn't quite the same thing. Again, Amanda Jorgensen is going to go through some of that information for grasshopper forecasts and the relative risk for next year, or this coming year, I should say. And then also some control products that are registered. So one other thing that we came to discover as part of our surveying work, this was not necessarily in canola, but we did actually come across cereal leaf beetle. And while this is not canola, while we were out at the same time, we have actually, again, been piggybacking some of our monitoring. And in fact, we have noticed there are more areas that seem to be showing both the cereal leaf beetle and the larval damage. So these larvae are very ugly, but one of the things they do very well is they're a defoliator that basically moves down the length of the leaf and chews out a very nice channel. And so cereal leaf beetle, even though we've seen it a few times, it's starting to now be more evident at more spots across the Peace River region. I bring this up because it's an insect pest that we need to keep closer eye on, and we're hoping to do more when we're out surveying for canola, at the same time walking across to the adjacent field and actually do more monitoring. Hopefully this is an insect that we're not going to see more of, but it does have a very broad host range. So it's a very challenging insect pest. The other thing I want to bring up here is that feeding channel. And one of the things that I just want to warn producers about is if we do actually happen to have a wet year, I have very much confused cereal leaf beetle larvae feeding for what looks like slug damage. It's an easy thing to confuse, and I bring this up only because a few years ago when it was wet, we saw gray garden slug creating the same channels. However, it's an insect pest that you have to monitor very late in the day for. In fact, at dusk is pretty much what you have to do. Some of the work we did with gray garden slugs, and we also tried to do a cereal leaf beetle, has also led us now into the realm of natural enemies. Okay. So with wheat midge, with some of our canola pests, we've now got projects where we're looking at natural enemies like these ground beetles. So we've been very fortunate, first of all, to have project funding to build capacity. We now have a technician in our program who is very adept at identifying different ground beetle species, and we're very fortunate that Shelby is working on some of these specimens because it's starting to help us understand what we're seeing in our canola survey samples, but also now with some of these other insect pests, some of which actually are very influenced by ground beetle populations, like slugs, for example. So some of this sampling is very difficult. It's pitfall sampling, which probably only an entomologist should ever try and do. It's pretty stinky and pretty finicky, but you can basically see we're taking samples that are retrieved from the soil surface. We're essentially bundling and then floating them in water so that we can pull out everything we need out of it. We start to build a sample at the bottom here that's organized by order, class, etc., all the way down to species level. And it's starting to be part of a larger Peace Region Living Labs project, in addition to what we're doing in canola and what we've been doing in wheat, in fact. So the diversity that we're starting to catalog in some of these pitfall samples is not only helping us understand what's going on at the ground level, it's starting to help us understand how what's happening in the canola canopy can be influenced by what's happening on the ground level. So I think one of the tremendous things with this table, we've done now 3 years of sampling as part of the Peace Region Living Labs project. This is only 53 sites in 2023. In 2024 and 2025, it's approximately 21 sites. But the tremendous number of insects in some of these commercial fields is actually very surprising to me, even though I knew there would be high numbers. The diversity and some of the densities have also been really surprising. So this is a really unique data set that we're anxious to do 2026 sampling in. But then to roll this all up is going to really be quite interesting to look at what kind of diversity and how much of an influence some of these insects are maybe having in our field crops. Hopefully what we're going to see out of this are some groups that are going to actually prove to be of economic value to you as growers. When I talked about Macrogleanes penetrans, that really small beneficial parasitoid wasp that attacks wheat midge, this is a very tight relationship that we understand with wheat midge. For some of these other species, like these ground beetles and rove beetles, we don't really have a good understanding of how closely they interact with some of our insect pests, nor how important they are to the overall field productivity. I do want to just emphasize that there's 330 taxon units in some of these samples. It's really surprising to see how many different things are active in your fields. And I think the more we understand, hopefully the more we will understand how to manage it well going forward. I just want to go through a few of these species here very quickly for you, because this first species of Bembidium is actually a very small beetle, but it's actually very much suspected that it influences and regulates pea leaf weevil simply because it's a predator of insect eggs on the ground surface level. This other beetle here, again a ground beetle, very colorful, 7 to 10 millimeters. It's supposed to be quite abundant on the prairies. We don't have a good understanding of what it may or may not be doing. However, it's got a long lifespan, 1 to 2 years. When we look at some of these other species, Poecilus leucoblandus is a carnivorous larvae. And in fact, the adults are supposed to consume some seeds, particularly weed seeds. And in fact, we find this with a few different species of carabids. They're actually living in and prefer weedy vegetation because they are either feeding on other insects that like the weed seeds, or they themselves are feeding on weed seeds. Pterostychus melanarius. Now, this is actually an invasive species, so this too was a bit of a surprise, although not astonishing. But essentially, we're finding a number of different species in here, different sizes, different lengths of lifespans, and different levels of maybe interaction, maybe functioning just on the ground level. Some of these as larvae, we don't even know what they do. And I think some of these large species, especially the Amara, this is a group that is well documented to feed on weed seeds. So even some of these ground beetles are performing some regulatory functions. So it's kind of neat to understand better what they're doing, what their role is, and perhaps maybe it gives us a better understanding of how we can augment or preserve them, particularly when we're not dealing with outbreak insect pest years. Well, I just want to finish out talking about some of the temperatures, because some of these insects, especially the invasive species, we know for our region that there have been changes. And so over the last few years, temperature has been a big driver for what we're seeing. And so this is actually kindly put together by the Prairie Crop Disease Monitoring Network. Sasha Chisholm and Kelly Turkington have pulled this data together. But for the Peace River region, we can kind of see what the average growing season temperatures were for 2025. It wasn't necessarily the hottest year in terms of average temperatures, but when we start to pull together temperatures and comparing averages over a 30-year norm, what we're starting to see as of 2025, sorry, can't speak tonight, is that we are actually seeing warmer growing seasons over the 30-year average. So anything in red here is actually 2.5 degrees Celsius warmer than a 30-year average. When we look backwards too, we're starting to see that not only temperature, but also, of course, our rainfall is influencing some of what we're seeing for both our field crops, but also our insect pests. And this is also contributing to why we're starting to see some of these invasive species. Things like wheat midge, like pea leaf weevil, probably. Cumulative rain, I mean, I don't have to tell you that we've had a few drier years, but when we actually look at the data for the percent of normal, it starts to be a little bit of an interesting story and reflects maybe how the piece is going to continue to change going forward. I do want to talk about pea leaf weevil. We definitely think that the temperatures and particularly precipitation have influenced greatly what's going on with this species. And in fact, when you folks download this, you'll be able to click on Kim Schorlemeyer's name. She very kindly recorded a pea field at harvest, August 21st, in fact. And what we were seeing were some very high densities of pea leaf weevil. Over the past almost 15 years, we've had pitfall traps out monitoring for pea leaf weevil. And one of the things we have discovered is not only do we know pea leaf weevil have been around Falher for several years, but we've also now detected them in some of our pitfall samples way out in the BCPs, particularly over here just north of Fort St. John. So here's an invasive species that's relatively new to our Peace River region that we now know that we have to start to manage a little differently. So things like some of those ground beetles that consume all of these pea leaf weevil eggs could be really more important to us in the future. I'm going to stop here. I promised you a QR link for our deck. I think the County of Grand Prairie is also going to be putting this up at some place, and there will be a YouTube video. I'm going to call it there and give you folks a chance to ask me whatever questions you'd like.
Thank you so much, Jennifer. That was really a lot of information. And we do have a question from someone here on the chat. Where has cabbage seed pod weevil been found?
Okay, if you'll just bear with me, I will scroll backwards and we will see across the prairies. So what I shared with you, of course, the deck is going to pause for us here. Okay, so this is the 2025 survey. So this is based on sweet mint collections across the prairies. And so anything that's highlighted as red, orange, or yellow, and even green, the dark green color here, this 1 to 3, that indicates weevils per 25 sweeps. So any of those areas across Western Canada indicate the presence of cabbage seed pod weevil. I do really want to point out a couple populations that have always been a bit worrisome, and that is just north of Edmonton, okay? And you'll see over in Manitoba, there is this population up here, which is absolutely confirmed as being a population of cabbage seed pod weevil. Those northerly populations of cabbage seed pod weevil give us pause for thought because we've always been concerned that they would cross what is essentially a forested area, what is fairly geographically remote in the Peace River region. However, despite the monitoring that we do here, particularly around Valley View, High Prairie, and throughout the rest of the piece, we've never been able to detect it. So I can tell you from our surveying, which ranges from about 104 fields to about 135 fields per year, we've never found it in the Peace River region.
Thank you. We do have one more question regarding the cabbage seed pod weevil. Has
Hmm.
the seed pod weevil's overwintering capability been studied, and do we know its ability to establish in the peas?
Yeah, so that's an excellent question. And yes, so when these first populations in 2003 were actually studied in southern Alberta, our colleague Dr. Hector Carcamo did a lot of work with cabbage seed pod weevil. First of all, looking at biology, looking at parasitism, and then also looking at overwintering biology. And some of the work that Hector found is that they don't necessarily tolerate a huge amount of cold. However, they overwinter underneath the snow in the leaf litter. And so their behavior and their adaptability actually is allowing them to tolerate some colder temperatures, usually around about 4 to 5 degrees Celsius minus. Those are negative numbers. And then beyond that, they can't tolerate it as well. But the important thing to remember is insects behaviorally find overwintering habitat. So there is the ability for them to adjust based on behavior, and this is always a concern. I just want to add, one of the things we were very hopeful about for pea leaf weevil, in fact, was that perhaps its overwintering biology would be somewhat similar to the cabbage seed pod weevil. And certainly it's proven that it's very different, simply because it's established up in the Peace River region now for several years, and we've seen it spread over to the northwest of Fort St. John. So every insect group a little different, but always keep in mind it's not just the threshold temperature. There's also some behaviors there.
Thanks, Jennifer. We do
Mhm.
have a few more minutes for more questions. So anyone online, please feel free to add them to the chat and we'll do our best to answer them. But otherwise, I'll pass it back to Kate. And thanks a lot, Jennifer. We really
My
appreciate
pleasure.
it tonight.
Yeah, thanks. While we wait just to see if we have any other questions, I'll just mention for anyone on the line that we do have Amanda Jorgensen speaking next week, as mentioned. So she'll be helping us learn about the pest risk maps and the pheromone trap results that are provided by Alberta Agriculture and Irrigation, and help us discover how we can use those tools, how farmers can use them in their operations. And then we've also reached out to Mike Harding, who will be recording a presentation. So he won't be able to present live, but he'll be recording a presentation on Peace Region disease updates and trends. So we'll have all of the videos available to people who have registered online on our county's YouTube channel. I'll email them out. And then anyone who's part of our Agriculture Monthly newsletter will receive them that way as well. Um,
If we
we have
have a moment, I do just want to say really quickly that I really want to thank all of the region's producers. We have a very cumbersome permission to enter form, and I've been so amazed and very grateful to the region's producers in that they've been able to take that form, give us permission, allow us to do some of this really important work. And so just a huge thank you to the region.
Awesome. Thanks, Jennifer. It looks like we do have one more question that showed up in the chat. So, do we know why M. bruneri in the BC piece is seen on even years compared to the odd years on the Alberta side?
Yeah, so that is an awesome question, and this is one of the things we really want to look at with the project. If you will bear with me, I'm going to scoot forward to some of those slides, if you will, because we didn't have time to talk about it, but it is something that we're very concerned about understanding better. Simply from the fact that when we talk about Brunner's grasshopper, sorry, how about I scroll and then here. Okay, so one of the things with grasshoppers, and when we actually look at this slide, so from 2023, and the data has been looking very similar across the last few years, these pie charts are various sites that we monitored in August. And one of the things that's very interesting to me, well, to all of us, it should be, The fact is that no two sites really have the same grasshopper composition for species. Now, every one of these sites has some of our native grasshoppers that are not economic pests. I just really want to emphasize that. However, one of the things we found is on Bruner's years, so this is 2023, it would be similar in 2025. This portion, this gray kind of browny color, reflects the proportion of the population at each site that was Bruner's grasshopper. So we don't have huge densities, but we do find Bruner's can be the dominant species throughout a number of sites. One of the things in the BC PACE, however, is that we seem to have these fluctuations of grasshopper densities almost on an annual basis and almost in some different areas. We're trying to get this type of information every year going forward so that we can better understand how much of those fluctuations in densities are associated with the Bruner's grasshopper. And so the interesting thing from 2025 is a couple of the sites that we actually went back to that were very high densities in July, we couldn't even find grasshoppers at in the BCPs. There were 2 or 3 other sites that weren't very high, happened to stop at in August as we were traveling through the region, huge densities. Those actually ended up being canola pellucida, so not even a Bruner's grasshopper. So this work really needs to be continued. We need to have a better understanding of the different species and how the densities are fluctuating from year to year. We suspect, or the literature, I should say, has indicated that the Bruner's grasshopper is supposed to follow a 2-year life cycle. So one of the things that we're doing, and I didn't have time to go through it, but one of the big reasons we're trying to collect these egg pods at the various sites in the piece is we're actually trying to pull apart the individual eggs and look at the development. We know for some of our grasshopper pest species that they typically will get to a certain stage, then overwinter inside the egg. However, their development through that fall helps us predict what kind of population or how soon they're going to emerge in the following spring. With Bruner's grasshopper, I don't want to jinx it, but some of the sites that we've been able to retrieve some of these egg pods, like here in this vial, what we're seeing, we believe that they're actually less developed than our more cyclical annual species. And this maybe then is why they're doing this 2-year life cycle. We have to get a lot more samples, and unfortunately, I'm almost hoping for some worse grasshopper years simply so we could get the densities to work with. But we need to figure out more about how those grasshopper eggs are developing and how they're moving on from year to year. So it's still a very big puzzle, and it's a really good question you're asking.